This is the big test, right? I've got all this information in. What do you think is the first thing I want to know? Yeah, all right. Well, and when I see what it looks like, what does that mean? Like, what am I looking for? Well, a factor. Well, so basically, the first thing I want to do is figure out if it's worth analyzing. So this is quality control effects. I want to see if this is even worth looking at. Here's where we do this, and I'm going to show you this shortcut for this. Workflows if you ever get lost doing, in Partech, go to your workflows. It is so simple, my high school kids do it. In fact, they'll be like, this is easy. And I'm like, yeah, I know. Go to gene expression. This will take you all the way through basically what we just did, except formatting the sample data. God, I wish there was a workflow for that. Okay. And again, you just it'll check the boxes off as you go through it. So now that we have the data in, now this second tab is quality control. Okay. Let me take a look at QC metrics. I can't remember what this one is. Oh yeah, it's not for Affymetrics. Here's what we can do. Plot sample histograms. Where is it? Oh, there it is. What do you think? So let me explain this. Actually, I have to explain it here because that's where my mic is. This is basically a histogram of the intensity of the signal. You'll notice that it goes from about a 0, actually it starts about 1, and ends up at about 13, right? Most genes are expressed in this area here. So basically this scale, when I see like numbers like 13, 11, 12, 8, 3, what they have done is they have basically normalized it using log 2. And that is something we do in bioinformatics all the time. Anybody know why you want to log 2 this stuff? Any ideas? No? What's the variability? So as you can imagine, I am surveying all genes in that mouse, every gene. And this is only like, epidermal stem cell, embryonic stem cells, right? Every gene is not going to be expressed. So I'm going to have values that are basically at baseline to like, say, albumin, which is like expressed at a high rate. So what happens if you don't normalize that data, you have a scale that goes anywhere from like 0 to 15,000. We don't like those scales in statistics. So if you take a log 2, basically what you're doing is you're reducing that standard deviation. You're smoothing things out. And you can do that because you apply it to all the data. You don't selectively do it. So here, if I'm looking at this, what we expect in most of these cells, even these embryonic stem cells versus the pluripotent, I would expect that most of those samples, most of the genes would be about the same. There's going to be maybe 1,000 or something that are going to be different, but it should look like they should all have kind of the similar pattern. When I look at this is telling me I have 2 populations in my group. I have these samples here and I can select them. I don't know why my thing won't let me do the box very well. So we've got this group here, which corresponds to this top group. So again, you can see here's all the embryos, or the cells from the embryos. So these are much different. These up here should be the same. You've got ESC cells, but you also have ESC cells down here. If you look, there doesn't seem to be much difference. Something's going on here. We have 2 distinct populations of cells, and you're going to see that once I do the PCA plot. And then obviously we have this group here. And when you put a box around something, oh man, I got to do it from a distance. That when you see this, that you've got two different populations, and what we're going to do is we're going to take a look at it on the PCA, and you're going to see exactly what I mean. All right. So everybody, so now let's go to the second one, principal component analysis.